All right, well, welcome back. And what we're going to talk about now is we're going to go. So we just talked about different types of cells and different cell types: prokaryotic cells, eukaryotic cells. And what we're going to do now is we're going to go through each one of those organelles, or those cell parts, and we're going to try to sort of look at what they do a little more specifically now. So we'll start by talking about cell membranes. And remember also cell membranes, just so you're not confused, if you see the word plasma membrane, that's the same thing. Cell membranes are what we call semi-permeable. And semi-permeable, permeable means you can go through, and semi means you can sort of go through them. So what happens is the function of the cell membrane is to regulate somewhat what moves in and out of the cell. So you might remember that in order to stay alive, one of the things that has to happen is living things need to maintain homeostasis. Homeostasis is the internal sort of balancing act that goes on. So the environment is changing. It gets hot, it gets cold, but as an example, as a human, your body temperature needs to stay sort of at about 98.6. So just an example. So 98.6 degrees Fahrenheit, which is 37 degrees Celsius. You got to stay sort of in that range, or as you know, if you get up to 101, 102, you start to feel bad. And if it goes down too low, like 95 or that kind of thing, then your cells aren't moving efficiently enough to do the chemical reactions that need to go on in order for you to stay you. So the cell membrane is designed to help regulate homeostasis and what can move in and out of the cell. So we call it semi-permeable. And you might have mentioned, might remember this from before, but we talked about the big main component of a cell membrane are the special lipids called phospholipids. And the phospholipids have what we call a polar head, and they have non-polar tails. These are the non-polar tails right here, and this is the polar head. So water can stick to this, but water does not stick to this part. So what happens is in a cell membrane, you'll form what's called a phospholipid bilayer. So the phospholipid bilayer is this double layer of phospholipids in which what happens is the heads, the phosphate groups, are pointed, say, to the outside of the cell, and another group of them, the heads are pointed on the inside of the cell. So you'll have water here, and you'll have water here, but you'll have the tails sort of turned inward. We also use the word, when we talk about polar, you'll see the word hydrophilic, okay, to describe the property of this part of the cell membrane. So hydrophilic, hydro means water, and philic means like loving. So we say that this loves water, and by that what we mean is that water can stick to it, okay? And then the tails in this region here, this region's called hydrophobic. Phobic, like phobia, means it's afraid of it, and so water doesn't stick to this. So people often sort of have the misconception that the water is repelled from it. And it's not that it's repelled from it, it's just simply that water has nothing to stick to here, so it will grab a hold of and stick to the phosphate heads here, but there's nothing to stick to here, so the hydrophobic tails sort of are just pushed in that direction, not trying to get away from the water. It's just a result of what happens as the heads stick to the water. And then inside, so it doesn't matter which direction you're looking at here, but in this particular case, we're saying this is the inside sort of portion of the cell. This is the cytosol, or the area called the cytoplasm, and this is the outside of the cell. And then we're going to talk about what moves in and out across that. So here's a close-up once again of the of the phospholipid, here's the phosphate group, there's the polar region there, and then there's the tails, and once again there's that double bond to have a bend in it, and so there's my phospholipid bilayer. There's other things in here we'll mention later on. We'll just start with the hydrophilic and hydrophobic regions though for now. Okay, so here's my cell membrane, here's my polar heads, here's my tails in here, so there's my phospholipid bilayer. You'll also see embedded among them, you'll see these proteins. Some parts of the proteins will stick out like that and will be hydrophilic. Some parts are inside the middle that are hydrophobic. The proteins have various functions. Sometimes they're used as cell recognition kinds of things. There's other things attached to it that allow your immune system to recognize, it'll bind to one of these parts and that'll recognize that that's your cell versus somebody else's or a foreign pathogen or that kind of thing. So we'll get more into that when we talk about the immune system later on. Once again, here's just another picture showing you the hydrophilic region, the hydrophobic tails. Now, when we talk about things moving in and out of the cell, one of the things we got to talk about first is we have to talk about what a solution is. So a solution is a mixture of two things in which you have a solute which gets dissolved, and then you have a solvent which does the dissolving. So what happens in a cell is like a bag of chemistry, if you will, and it's surrounded by other bags of chemistry and other fluids around it. So the solute is the solid stuff usually. So if you take salt, for example, and put it in a solution, it will dissolve, and the solvent in living systems is usually water, but you could have other solvents in, if you're doing industrial chemistry stuff, you might have a lipid of some sort or an oil that's your solvent. But in living systems, we're talking about water as the solvent, and then the solute is the other stuff. And what happens is we use terms to describe one location versus another. So we'll use the word hypoosmotic when we talk about a solution that has less solute than something else. This is a little bit tricky because the word osmotic, we'll see, is often just related to water. But in this case, when we talk about hypoosmotic, it's important to note we're meaning less solute compared to something else. You'll also see the term hypotonic instead, and some people prefer that word better. They have slightly different meanings in this class. We won't worry about that. Hyper means more, iso means the same. So hyperosmotic or also hypertonic is going to be used to infer that something has more solute than something else. So we have hypo meaning less and hyper meaning more. Iso means the same, and once again you can have isotonic as well. So we use terms to describe one part compared to another. So for example, in this case, let's say you have a cell here and you have the outside of the cell, you have 10% glucose, on the inside you have 3%. You would say in this case the inside is hypoosmotic to the outside. And in this case over here, if they're both the same, they're isoosmotic or isotonic. And if it's 50% glucose and then 10% glucose as an example, then the inside of the cell is hyperosmotic to that. I don't know if I said that correctly before, but just in case, here we have hypoosmotic, less, so there's less solute than this one here. And then if I asked you, just to, sort of ask you and see if you can, if you're thinking about this, okay, what is the percentage of solvent, say in this cell right here? So if I asked you what's the percentage of solvent, okay, well, if we have 3% glucose and then we know that everything else is the solvent, then the solvent concentration in there would be 97%. Okay, just like in this one it would be 50%. Okay, because 50 plus 50 would equal 100%. Okay, all right. Now, things that can go through the membrane versus things that cannot. Okay, so it turns out because of their size, first we'll talk about things that can go right through the membrane. So talking about molecules that can go right through that plasma membrane. If they're small and they're uncharged molecules, which means they don't have a negative or positive charge to them. Like if you look down here, sodium and potassium, these are like ions or electrolytes, and those cannot go through the membrane. But CO2, for example, uncharged, oxygen, they can go right through the membrane. Things that are small and polar, okay, so they have a little bit of charge to them. You know, water sticks to itself by those hydrogen bonds. Water can also go right through the membrane as well. Even things that are fairly large, if they're non-polar, so they're lipids, things like steroids, can go right through the membrane. So cholesterol, for example, although it's a big molecule, when it's turned, it's kind of flat. It's like a piece of paper, sort of. So even though it's large like that, it can turn sideways and it can go through the membrane. Okay. Things that cannot go through the membrane are charged molecules like ions, sodium, potassium, chloride, for example, magnesium, okay, can't go through. So several of these cannot. There are ways to get them in. We'll talk about that, but they can't go right through the membrane. The reason they can't is water will bind to these and prevent the water from moving around. So you have water inside the cell, water outside the cell, and so it's very difficult for the sodium to gain enough of a pull away from the water to get through the membrane. And large polar molecules also cannot go right directly through the membrane, such as glucose. Okay? Now, when we talk about things moving in and out through the membrane, there's different forces sort of involved. And first we'll talk about passive transport. And passive transport comes in several different sort of styles, but simply what happens with passive transport is you go from an area of high concentration to an area of low concentration. And this can be just through random molecular movement. Okay, so let's say we have, in this case here, we have a bunch of water on the outside of the cell, and on the inside we have a little bit of water. Okay, well, water, if it's just moving around randomly, and you have more on the outside versus the inside, the chances that one of these water molecules here is going to move across the membrane is going to be much, much greater than the chances of one of these moving across this way, because there's so many more out here. Okay, so although water, if it's in high concentration here versus here, although it can move both ways, and it will, what you expect is at the area of high concentration, more of the water is going to move this way. So we're going to get a net or overall effect of water, in this case, moving from the outside to the inside. And it doesn't really matter if it's outside or inside. If you were to reverse those and you had this area in here, it would still go, for the most part, from high concentration to low concentration. So that's called simple diffusion. Simple diffusion is just simply moving from an area of high concentration to an area of low concentration. Now, if a membrane is involved, this is kind of a term you don't see very often in the books anymore. We use it in labs, so I'll point it out here. Dialysis, and you do actually, hear about this when you think about kidney dialysis, which is kind of what was going on here. Kidney dialysis, or when you talk about dialysis, you're talking about a membrane being involved. So simple diffusion can just be a gas going from one part of the room to another. So if you have if you spill a bottle of, I did this, my wife's perfume, I was reaching up in the cabinet, knocked down the bottle, and broke, her whole bottle of perfume all in this bathroom here, and it was like all over the floor and everything. Well, it's at high concentration there. You can smell it in other parts of the house as it diffuses from high concentration to low, but the majority of it is right there. If you involve a membrane where it has to go through a membrane, then that's called dialysis. Okay? Now, if what you're talking about is specifically water, then you're talking about osmosis. So osmosis is the special case of diffusion or dialysis but specifically with water. Okay? And then facilitated diffusion is what you see over here, still going from high concentration to low, but what happens is there's a protein involved or some other mechanism to allow it to get through the membrane. So there are these transport proteins that can often be opened and closed, and we'll talk about that when we talk about the nervous system. But in this case, this is facilitated diffusion because the molecules have to travel through the protein. They can't go right through the membrane. They have to go through the center of this protein. And those are transport proteins. Okay, so those are all passive transport. They're passive because they do not require energy. They simply go from high concentration to low. I often equate this to like, this is like a rock rolling down the hill. All of these are like the rock rolling down the hill. They go from an area of, so if you draw a hill like this, okay, and you, oops, I lost my hill. Where did I go? There we go. Come back. Nope. Yep. Here we go. If I have my hill, okay, so there's my hill like that. If I have the rock up here, passive transport is the same idea as this rock going down the hill. Okay, if you just move whatever out of the way that's holding the rock, it'll probably roll down the hill. But what if you want to move the rock up the hill more? You're going to have to put energy in. The rock's not going to go that way on its own. It requires energy to get it up the hill. And sometimes you might have a molecule in one concentration, and maybe it's already at a high concentration, but because of the biochemistry of the cell, it's required that it's at a higher concentration. So you need active transport to do that. And so what happens then is you use energy up to do that. One of the most common sources of energy, I don't know if we've hit this yet or not, but ATP. ATP is a very energy-rich molecule. It's not the only one, but often when we see reactions that involve energy, you'll see an ATP and an arrow going into it, implying that there's energy that needs to be put into that situation to make it happen. Okay? So what happens here is, let's say you have high concentration here, low concentration here, but you want to move these few molecules this direction. In other words, you want to make it even more concentrated. Passive transport, they would flow that direction overall. Okay? But if you want to move more of them in, you need to use energy to do that in the form of ATP. One example of that, so this is just an example here, is the sodium-potassium pump. Okay? And you might wonder, what's the sodium-potassium pump do? Well, luckily when they named this kind of thing, they made it nice and easy. It pumps sodium and potassium. That's what the pump does. So it pumps sodium and potassium. And if this is a neuron, for example, or a nerve, what will happen is in order to make the cell electrically charged, there's a special kind of protein that will pump sodium out and it'll pump potassium in. And it's active transport because regardless of the concentration gradient, the neuron, in order for it to work properly, needs more sodium outside and more potassium inside. We'll get to that later. So active transport, this is an example. I just want to make sure you know this is an example. Active transport means it requires energy to make it happen. The sodium-potassium pump is an example of that. Okay. You often might need to move large things in and out of a cell. And so we have ways to do that too, called endocytosis and exocytosis. So what will happen is, if you look at this picture here, let's say you have a whole group of molecules or proteins or whatever that you want to get on the outside of the cell. They will be wrapped up in this membrane type of material, and then what will happen is that will fuse to the membrane and you can dump that out. You'll also see that's exocytosis. So exocytosis is moving things out of the cell. Endocytosis is moving things into the cell. So let's say these are bacteria on the other hand, or whatever it is that you need to get on the inside of the cell. Certain immune cells will take these bacteria bind to them and will engulf them. It'll form a membrane. And if you go this other direction, you get the same idea. Then you have all your things that you were trying to bring into the cell enclosed in this membrane. You can fuse that with a lysosome and digest that if it's bacteria, for example, and you can destroy it. When you talk about endocytosis, just since we're here now, you'll often also see the term phagocytosis, or the term, not as common, I don't think it does not have a H in it, pinocytosis. So pinocytosis is when you're bringing in water. So the endocytosis of a liquid, I should say. Any kind of endocytosis that involves a liquid, sometimes they'll refer to it as pinocytosis. That term you won't see as much, but phagocytosis you will. So phagocytosis, I should mention here, is a type of endocytosis, particularly of solid organisms. So you'll see phagocytosis, for example, of bacteria by your immune system. Okay, just an example of endocytosis. Okay, and we have other things attached to the membrane as well. We'll just kind of mention here briefly so that you have an idea of them later on. There's carbohydrates that are attached to those phospholipids. We'll call those glycolipids, for example. So those are lipids, phospholipids that have the glyco part is basically the carbohydrate part. There's proteins. There's also proteins with carbohydrates attached. So those are glycoproteins. Cholesterol. So there's I want to get just, you don't need to worry too much about this right now, but the idea is that in addition to the phospholipid bilayer, there's also these other molecules, and they're important for a variety of things such as cell-to-cell recognition and that kind of stuff. The cholesterol is important to keep it fluid. So we often call this model, when we describe the membrane, we call it the fluid mosaic model. It's a common term. There's some new research out that is suggesting it might be more fluid than we think, but for a long time, this has been sort of the standard model of describing a cell membrane, because it's not a solid structure. It's sort of moving around, and there's proteins there that come and go and are kind of sloshed around in this solution, if you will. But your cells, for example, in your body are designed to recognize each other. And if there's a foreign invader, for example, if you get infected with a bacteria and you don't see these certain markers on the cell, these certain glycoproteins, for example, your immune system is designed to destroy those invaders. Could be your own cell even. So, for example, let's say you get sick, you get a cold. The cold is a virus, so it will infect your own cells, and what happens is your cells become damaged, and as they become damaged, one of the things that gets messed up is in your own cell that's infected with the virus is this cell recognition. So your immune system can recognize your own cell, but that your own cell's damaged, and it will destroy your own cell so that virus can't spread. And that works really good, really well when you have the cold. But the problem is there are a few diseases, like, for example, HIV, in which HIV tends to infect your T cells, which are one of your immune cells. And so what happens is your immune system does what it's supposed to do. It recognizes that it's your own cell, but it's infected with this HIV virus. And so your immune system tries to start getting rid of and destroying your T cells, and that wipes out your T cells, which is the problem with HIV. So here's just a picture showing you the different kinds of things, like here's a glycoprotein, for example. Here's a protein with a carbohydrate on it. Here's some cholesterol inside here. So even though you think of cholesterol as being bad, you actually need some cholesterol in your cells. So low cholesterol levels are also problematic for cell function. If you live in the United States, we usually don't have a problem with our ability to get cholesterol because of our diets and so forth. But that could be potentially a problem if you decide to like have no cholesterol, for example. So I think that's it for now. It gives us a nice tour of the cell membrane, and we're ready to move on next to talking about energy.